Protein synthesis is the process of making protein, and we need proteins. Why? Because proteins are the executor of cell-life activities. We are made of cells, so for us to be able to function properly, our cells must be able to produce proteins. And remember, proteins are made up of amino acids, and amino acids are the building blocks of protein. Without these amino acids, there will be no proteins. There are three steps of protein synthesis. First is the DNA replication. DNA replication is the biological process in which cells create two identical copies of DNA from the original DNA molecule with high accuracy.
When DNA replicates, its strands are separated by the enzyme helicase. Single-stranded DNA binding proteins keep the strands from re-annealing. One DNA strand encodes the leading strand, which forms from its 5' to its 3' end, using DNA polymerase III. No problem here, but the lagging strand presents problems. It has to form from 5' to 3' too. It forms in pieces called Okazaki fragments. First, an RNA primase lays down an RNA primer. Then, DNA polymerase III lays down new DNA. The process repeats again and again. DNA polymerase I replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragments.
Now let's go to transcription. In eukaryotic cells, transcription occurs in the nucleus, where the DNA is used as a template to make messenger RNA. During RNA transcription, a region of the DNA that was replicated a while ago will be converted to mRNA. M in the mRNA stands for messenger. And lastly, let's go to the third process, which is the translation, which occurs in the cytoplasm of the cell. The information contained messenger RNA is used to make a polypeptide. So what happens during translation? So what happens is that information in the nucleotide base sequence of mRNA is used to dictate the amino acid sequence of protein.